I'm Harry Glurkian, and this is Moneyball Medicine, the interview podcast where we meet researchers, entrepreneurs, and physicians who are using the power of data to improve patient health and make healthcare delivery more efficient. You can think of each episode as a new chapter in the never-ending audio version of my 2017 book, Moneyball Medicine, Thriving in the New Data-Driven Healthcare Market. Matthew, welcome to the show.
Oh, thanks. It's good to be here.
So, spend a lot of time reading about what you're doing, your past, and sort of the history here. But I want to start off with which fits right into the show is you've said that data is the greatest drug of the 21st century, and that precision medicine delivers data as a drug. Can you expand on what you mean by that statement?
Yeah, and I've said this a few times in a few ways, but the principle here is that I think we need to look at data itself as a kind of intervention. That exposure to one's own data could have ramifications for your health. And you can imagine this in a very general sense. Like if you get detailed data about your health, you might do something about it. Right. But if you give extremely detailed data to your physician, they might be able to do something with it. And oftentimes I'm thinking in terms of the very molecular in that case. And that's really where I spend a lot of my time. But giving clinicians molecular resolution on the nature of your specific health problems is really what I think is so revolutionary about medicine right now. And the ways in which we can gather that data and the computational tools that will be available someday to physicians, and even to an extent right now, will enable them to do things that no drug can do on its own.
Well, it's interesting that you say data, and in that sense, is I was just playing with something that pulled in all my medical data and put out all the longitudinal charts for me and highlighted all the ones where I was out of whack. And I texted my doctor, I'm like, we need to get on a Zoom call. I need to show you a couple of things that are out of whack, and I want to figure out why they're out of whack. So I agree with you that molecular will be that really high resolution level to get to, but even I think the simple data to give patients, I think, is powerful. It can move the needle if we can communicate it to them effectively.
Yeah, I think even the simplest incarnation of this could do some good. Like imagine if your scale not only told you your current weight, but just drew a line between your last 3 readings and said, this is where you're going to be in 10 weeks. Something that simple might help.
My scale does do that, and it tells me that you are getting fat, so you need to do something. So I try to intervene when I can. But so you have an interesting history and past. I mean, you went from pure computer scientist from the University of Utah into precision medicine at the University of Alabama. I mean, and I almost want to say, based on what I was reading, like that revolves around your personal experience from finding a diagnosis and treatment for your son, who has a rare genetic disorder that sort of went undiagnosed for 4 years, I think it was, if I remember correctly.
Yeah.
Can you give the listeners maybe a condensed version of that story? And
sure,
how did it turn out that your experience studying things like functional programming turned out to be so useful for studying rare diseases?
Yeah, well, that's a wonderfully broad question, and yeah, I've had an unusual path to this point. So I'm currently the director of the Precision Medicine Institute at UAB, and so it's a very focused medical research institute. We want to help patients find tailored therapies for them. My background in computation and computer science certainly influences that. We have a host of computational tools to help do that. Some are based on artificial intelligence. Some are very systems biology focused that start to invoke aspects of functional programming. And you're right. And the reason I started all this is my oldest son, who unfortunately passed away in October, had an undiagnosed disease, and for 4 years had no idea what he had. Eventually, through a novel application of exome sequencing, was able to determine that he had the first case ever known of this ultra rare disease called NGLY1 deficiency. And I think that's safely the point where I really flipped in my head from a computer scientist to an amateur biologist. I knew enough to try to get him diagnosed, but that's the point where I said, I've got to find some way to help him. And even though he passed away, it's hard to look back on his life and see it as anything but a major success in many ways, because he was born with a very short life expectancy, and yet he made it to almost 13 years old. And that would not have happened without sort of a sequence of emerging technologies that came just barely in time to extend and enrich his life and bring him a lot of joy. And so I miss him every day, but I'm lucky that I have the opportunity to work every day, literally every day, towards his legacy of helping patients with science, a lot of which is computational, but much of which is sometimes just good old-fashioned wet biology, where we go to the bench and try something out.
Yeah, I mean, you've sort of described the Precision Medicine Institute as a form of research consultative service, where the goal is to find the next step for any patient that reaches out on their diagnostic or therapeutic odyssey. I mean, that sounds amazing. I mean, maybe you could sort of describe more of what happens on a day-to-day basis.
Yeah, so it all comes down to Monday. So Monday is case review day. So if someone has reached out to the Institute, or if they're sort of currently working on something for them, Monday is where we all synchronize and put our heads together and try to figure out that next step. So for patients that have reached out for the first time, it's okay, what's the first step? Yeah, in the direction of a therapy. And for those that are in flight, if there's been a change, if some experiment has completed, if some lab has come back, if new information has been introduced, we check to see, is there a new next step? Is there some new insight? And sometimes on those Mondays, information will come back that enables a query to be run in one of our computational tools, where the best example would be a target's emerged. Like, if we modulate the behavior of this gene, we think it will be therapeutic for this patient now. And so we can run queries to see, can we modulate the behavior of that gene using some kind of small molecule or some other approach?
So, but when you're doing all this, I mean, do you have a mission to sort of either scale up or automate? And if you do, how is that going?
Yeah, so it's both, and in fact, it's scaling through automating. We realized pretty early on that humans are an essential part of this process right now, in the sense that humans really do need to. In this case, when I say humans, I mean undergraduate students, because they're the ones within the institute that act effectively as the case managers and reach out and sort of pull in the information, digest it into some kind of structured format that the tools can process. They might engage with the physician. They might reach out to some basic scientists that have insight on the relevant biological processes and figure out, how do we drive it to a query or a recommendation for an experiment? And so in some sense, when I think about scale, what I'm really thinking about is the efficiency of these undergraduates. How many cases per week can we get them to process, and how much tooling and automation can we build to make them better and better at what they do? So that's how I think about scale. And I think about replicating this as a process in every academic medical center across the country. There's no reason you couldn't have a team of extremely bright students at every center in the country that run this kind of process for their patients locally.
Yeah, I mean, I would think that would be one hell of an experience for the students to sort of see something actually being practically applied, as opposed to reading it in a book and it being a little bit more theoretical.
It is actually. In fact, I've noticed that literally 100% of the students that have participated in this program, I mean, all of them, have gone on to graduate school either for an MD, a PhD, or both. So it's a 100% success rate to getting students into grad school or med school. And now we have a course version of it. And so in fact, several course versions of this, where you can take this class and you'll get to practice on some existing solved cases, but we even throw some unsolved ones in the mix to see how they do when they take these courses. And then for the honor students now at UAB, they can take a special course sequence as freshmen, where they'll get into the lab and build model organisms that represent some of the patients we work with. which could ultimately enable the discovery of therapies for them. So I have to say, I can't think of a lot of other places where you get that kind of experience as an undergraduate.
Well, no, that's what I was thinking. I was thinking, how do we get this more broadly out there so that more people are doing this and get their head in that zone and understand these issues? But I think one of the projects that I was looking at was Medicanron.
Yes.
Is that, if I'm pronouncing it correctly? What is it meant to do?
So Medicanron is really our flagship artificial intelligence tool that we use primarily for drug repurposing. That's what we kind of built it with the end application in mind. So I'll tell you what it's really good at doing now. If you tell it a gene and you tell it a direction to go, like I want to make this gene more active or make this gene less active, it does very well on those kinds of questions. And it can scour a number of data sets to do this. So we're part of actually a larger effort through the NIH called the Translator Consortium. This is a huge research effort. We have lots of teams working together to both mine out all biomedical knowledge and make it structured, and another set of teams which are trying to do automated reasoning on top of all of that knowledge. So we're on the automated reasoning side. We can do some of the mining too, but the other teams do such a fantastic job that we mostly just consume what they produce in terms of the mining. And then we try to stitch it together so that we can find interesting ways to go after targets of interest for our patients.
So, it's reasoning, over medical knowledge graphs, I think, that you're trying to do. And so it sounds like a promising way to find unexpected connections between, say, disease and existing drugs. But if you had to explain that to a layman, how would you explain sort of, a knowledge graph and what you guys are doing with it?
Sure.
Or do you have a favorite example?
Well, I can, I have pictures I can do with, but I can also do it with words. So, a knowledge graph, let's talk about what it represents. Ultimately, there's a structure, but that's not actually all that important. A knowledge graph is a collection of facts. And facts are sentences. And they're sentences of the form A somehow relates to B. So in some sense, a knowledge graph is just a huge collection of these sentences. A is related to B, where there's a specific relationship. So a biomedical knowledge graph is going to have some constraints on it. So the A's and the B's that you're connecting are going to be nouns from medicine and biology. So there'll be drugs and diseases and genes and metabolites and all the other stuff that you typically read about in medical papers. And the relationships are going to be biomedical in nature too. So it's going to be things like A inhibits B, A activates B, A causes B, A treats B. And so if you collect all of these sentences together, you have what we call a knowledge graph. And the cool thing about a knowledge graph is that you can do logic on top of it and try to look for relationships that are there, but not explicitly stated. And then the simplest example of this is, let's suppose there's two sentences in this knowledge graph. There's A increases B and B increases C. We can infer logically that if you increase A, you should also increase C, because B went up and so C should go up. So that's an example of logical inference on top of one of these knowledge graphs.
And so that's typically, there's a human intervention at some point to sort of look at this and then say, yes, this makes sense.
Yes, absolutely. So one of the major roles of these undergraduate analysts is to actually double check what comes back from a tool like this, because it's going to emit a logical argument. It's going to say, I believe that this is going to influence the right target because, and then the analyst can look at the because, and it's going to have references into biomedical data sets. It's going to have references to papers in PubMed, and they can go read those. They can look at the data sets and they can double check the reasoner and say, you know what, I think you got this right, or no, you made a mistake. And it does make mistakes sometimes, so a lot of the knowledge from the literature has been done by natural language processing, and that makes mistakes. Right. So I'd say it's critical to have a human in the loop to double check that. And towards your earlier question about how we do scale, one of the things that we've added to the tool is ways to make that check go faster. So for example, in the latest interface, when it tells you that it believes, for example, A increases B, and you click on that, it's going to jump straight to the sentence that it pulled that from in the paper. And so you can just look directly at that sentence and say, do I believe this? Do I believe it got A right? Do I think it got B right? Did it get the relationship right? It's sitting right in front of the analyst, whereas previously that was a few clicks away. They had to, click on that. They had to click on the paper it found. They had to go to the paper in their web browser. They had to look at the abstract. They had to find the sentence that it got it from, and then figure it out. That's a long process, actually, as it turns out. And going from a few minutes to verify an inference to a few seconds, that's a huge increase in efficiency for these analysts.
Well, I mean, if one of the things I would say is I always try to find out, like, if it shortens the overall process of even finding this relationship. I mean, if you had to put sort of timescales on this, is how much faster do you think that we're speeding up this whole process of being able to even identify something that might have this effect?
Yeah, I mean, we've had some natural experiments in this regard, where in some cases there were answers sort of buried in the literature that seemed to have been therapeutically relevant, and yet very motivated disease communities hadn't stumbled across them, and motivated physician scientists researching these diseases had not run across them, or didn't sort of connect the dots to realize that this could actually be relevant to a patient. Probably the most recent example of this is ADNP-driven autism, where there were results in the literature that could imply the key finding, which is that low-dose ketamine will increase ADNP. That's the key thing that the researchers trying to treat this disease were after. And in some sense, that was out, publicly known, if you will, in the literature. And yet it took, running this query to find it, to sort of make the realization that this was true. It's kind of interesting, actually, that to think about the fact that there's stuff out there that as a species we know, but we don't know that we know it. So we call that sometimes the unknown known.
Correct. Exactly.
It actually happens a lot in different contexts. I even remember this happening in computer science, where there were communities out there so disparate that one had solved a problem the other had been trying to solve for 3 decades. And they just didn't know that the problem had effectively been solved. I mean, it can happen, actually. Science is just so big.
Yes, and I talk to different groups that are working on systems that will make those unknown knowns more, easily findable, or at least highlight them so that people know that they're. But you guys search scientific literature, drug databases, for existing and approved, and basically you're looking to find something that's going to perturb a ultra rare disease. Why is it better to look for an existing drug rather than a new one? I just, curious of the practical arguments around that.
Yeah, well, it's not, I'm not against developing a novel drug for a single patient. It's just that most patients don't have $2.6 billion. So it's a little out of their price range. That said, of course, there's technologies that are changing this equation substantially. So I would say oligonucleotide therapies in general, it's not down to, $1,000 a patient, but it's dramatically less than 2.6 billion. We're probably closer to the range of a couple $1 million, and that's falling fast to do these sort of custom programmed therapeutics for individual patients. So, I'm not against finding novel matter. It's just that it's still outside the budget of what most ultra rare patients can handle.
Right. Well, I was talking to, actually, it hasn't even come out yet, but I was talking to Kevin Davies in one of my last podcasts about CRISPRs, and just exactly that same discussion. So I don't know if you guys have sort of done a ballpark or sort of a thumbnail of, what do you think, what fraction of rare conditions do you think, are treatable the way that you guys are approaching it? Is it fair to say that eventually you'll exhaust that approach and that we'll have to develop a new drug for,
Yeah, and I guess we got to sort of clarify, what we mean by approach. So there's the AI-based approach, or sort of strictly computational approach. And then there's what we can do if we're allowed to go to the wet lab for a little bit of stuff. And if you play the game where you can only use a computer, The answer, there's the answer for today and the answer in the limit. Once we sort of saturated biomedical knowledge graphs, if you will, with everything we're ever going to know. And, already, right off the bat, I think we jumped to a reasonable suggestion somewhere between 5 and 10% of the time. This is for the case of ultra rare genetic diseases. And there are factors that can influence that. So, for example, if it's a dominant disorder where the genetic insult has really just sort of tweaked the thermostat on a gene, so it's a little overactive or a little underactive, we tend to have a better success rate jumping straight to an answer with a computational tool than if the gene's been wiped out. And now we have to find a way to replace that activity. Now, the good news is, if you look at sort of the census of rare disease, 75% of all patients fall into that bucket of the gene's become a little overactive or a little underactive. And so it's very amenable to an approach like this. And for the patients that, where the gene is missing, there are still things we can do computationally. The call I had right before this one was exactly that case. What can we do computationally? And by playing with the tool, we found some alternate targets to go after. But it takes some play to do it at that point. It's not quite as automated, but you're still using the tool as targets emerge to ask the right questions.
Yeah, when I think back, like, what you're talking about would have been dreams, 10, 15 years ago, right? Of what we can do now computationally versus what we could have done back then, and the information that's available because of genome sequencing and other technologies that have made it available. But before you moved to UAB, you were working on the precision medicine initiative for the Obama White House, I believe, and then briefly for the Trump White House. Can you update on the status of the, was it All for Us initiative, and then Patient Empowered Precision Medicine Alliance?
Sure. So yeah, I did spend, I think in total, about 3 years either working with or working for the White House, under both Obama and Trump, on the Precision Medicine Initiative, related initiatives like the Million Veterans Program, little micro initiatives like the Patient Empowered Precision Medicine Alliance, where we're just kind of trying to test it out to see if these ultra tailored approaches would work. So as far as where things stand today, All of Us is a very successful large-scale clinical genomics research program, which is, I think, on the way to hitting its target of enrolling 1 million Americans. And the way I described that program, even at the time, was like it's like you're trying to build the Rosetta Stone of the human genome. It's turns out we reached a point where it's really easy to sequence a genome, not so easy to interpret a genome. So if I sequence you or me and we find mutations, we go, well, that's interesting. What does that mean? We go, I don't know. But let's suppose you got 1 million people to donate their health records and their genomes. Well, now you can start to draw, statistical connections between what this mutation or this collection of mutations means in terms of actual human health. So it's finally a way to start decoding it. And so that's really what All of Us is about. In my view, it's about building that Rosetta Stone for the human genome. And then, for the Million Veterans Program, it's actually sort of inside of it, yet also outside of it, and really trying to do the same thing, but leveraging the extensive, clinical records and histories that the VA happens to have access to, and taking a slightly different method to get there in terms of genomic data, starting with, SNP chips and genotyping as opposed to full-on sequencing. And then for PETMA, it was really a pilot project where we just said, okay, can we take a small number of patients and can we actually sort of run this process all the way forward where we get a genotype and try to find a medication that might help them? And it turns out for a handful of cases, we were actually able to do that. It was all thanks to, getting a group of, private entities like companies and universities to come together, so that we actually had enough infrastructure in one place to run the process. And so we were actually able to do that.
Yeah, it's funny because I always think to myself, like, all these silos, if we could just have them integrate in some way, you'd have a lot more data to work with and find. I always find that in the beginning, you always find those low-hanging fruit that sort of fall out, and then it gets harder. But if you have enough to start with, something interesting falls out of it.
Yeah, yeah.
So you said in precision medicine, for a lot of cases that we deal with, we don't have sort of the right drug right away, but we can always prescribe an experiment. So what do you mean by that?
Yeah, by that I mean, and so this gets into sort of like the philosophy of medicine itself. So you'll hear clinicians use terms like this is not actionable. Yeah, and then you hear that an awful lot in rare disease. You hear it, a lot at the end of cancer, where they'll say, "Oh, there's nothing we can do," or "There's no clinical utility in this." And I think precision medicine subverts that whole approach and says that, well, if you've run out of clinical options, you can still do some science. And increasingly, I think we can systematize that science, so that it's not, okay, we need to do something. It's here's a set of things that you could reasonably do at this point, a set of experiments that, if run, might point to what to do or might point to another next step. And a great example of that is, for particularly for a rare genetic disorder, in many cases, you can build a model organism. You can build a fly. You can build a worm pretty inexpensively, and to model that genetic disease using things like CRISPR. I mean, people don't appreciate, I think, the full importance of gene editing. I mean, people think about editing human genomes, but the fact that it made it so much easier to edit animal genomes was actually, in many ways, an even bigger impact, and a more immediate one as well. So yeah, you can, for virtually any genetic disease, if there's an animal equivalent to that gene, you can build the animal. And if it's a small animal, you can test a lot of drugs on it pretty inexpensively. So I've got a friend and collaborator, Ethan Perlstein, who built a company around this approach and was very successful actually in treating some patients this way. I have a collaborator, Clement Chow, who's an academic doing this on the academic side, focused on Drosophila, doing this very successfully. So, that's not, a drug. It's not a procedure, not a medical procedure anyway, but it's a very well-defined process. And it's a process whose outcomes could be measured statistically. So you might even know what your odds of success are before you try it, whether or not you're going to find something. So I think it takes an evolution in our thinking to realize that this is a perfectly reasonable thing to do for a lot of patients. Build that fly, build that worm, and test a bunch of drugs.
There's a lot of times where things seem perfectly normal for me to suggest, and I've had people look at me like I just grew like 2 more heads off my shoulders. So, but this sounds like this is your fundamental belief that there is nothing that is not actionable, medicine or otherwise.
Right. Yeah. I think it also requires a degree of stoicism, because just because there's something to do, it doesn't mean it's going to work in time. And this is something I was always mindful of, during my son's life, was that while there was always something to do, I was mindful that it was probably not going to always happen in time. It was always a race against the clock, but there was always something to do. And even today, there still is. I'm still working on his condition. I'm still very actively engaged in drug development for his disorder. So even now, there's something to do, and as a parent, it still brings me benefit to know that it will benefit others. So it does require a shift in perspective about the meaning of actionable.
Well, so it's interesting or strange to say, but it feels like finding ways to use existing drugs to help people with rare problems is waiting to be solved with someone with your exact skill set in computer science and your exact set of motivations as a father of a child with a genetic disorder, right? And so many other key players that I've talked to in this sort of, that have this N of 1 stories, have very similar biographies. I mean, there's been, a few movies made about it, right? It makes somebody wonder that if your son hadn't been born to you with your expertise, that could apply knowledge and that experience to it, that you wouldn't be moving the ball forward. How does that make you feel about the state of science or medicine?
Yeah, and you're not the first to make an observation that I sort of ended up in the right place at the right time with the right set of motivations, and there's a lot of truth to that. I think about if he'd been born even a few years earlier or a few years later, how things would have played out differently. And yeah, I realized early on that there's a desperate need for computer science within medicine, that there is so much opportunity just left on the table for lack of expertise. But I made a deeper observation, which is that as much as medicine needs computer science, what it needs even more is computer scientists. Now, the problem is that the average computer scientist doesn't have sufficient motivation to go learn medicine. Medicine's big. And
Yes.
biology is messy. And I got to say, biology is so messy that to the mind of a computer scientist, at times you're just like, God, what a disaster biology is. It's like every time you have a rule or a law, you have to put probably or most of the time at the end of it. There's nothing ever that is just always true. And when you come from a field like computer science where you can put clean little theorems around everything and layers of abstraction that never break, it's like, oh gosh, who designed all this? Who's the engineer that thought that was a good idea? That's how I feel half the time when I dive into biology. And yet there are abstractions that you can borrow from computer science, and you can use these things to start to describe the way biology does what it does. And so I do think of the cell as a computer or a machine, maybe a Rube Goldberg machine, but a machine nonetheless, and one that you can sort of intellectually approach, from the direction of computer science. And within computer science, I happen to have a background in functional programming. And there are times when, describing the mechanics of how biological processes operate, where I'm like, gosh, this kind of feels like I'm playing with a little functional programming language, like I'm doing graph rewriting instead of term rewriting. But it's, there's been these moments where I'm like, yeah, this is just a programming language. It's weird, but it is one. And so and then I think, gosh, I mean, it is strange that I arrived with that particular skill set at this point in biology's history to make these observations and use that profitably towards helping patients. So in terms of how that made me feel, lucky, I guess, that I've just sort of been there in the right place at the right time. And the same thing is true for almost everything else that's occurred, since Bertrand was born. From the timing of the Precision Medicine Initiative itself, to getting his story in front of President Obama at just that moment, to getting the invitation to go, to getting the invitation to participate, and then join the team. I mean, the timing and all of it was just so ridiculous to me that I look back and think, I can't believe that happened.
Right. Wow. I mean, timing, being in the right place at the right time, a little luck. I'll take that every day, right? Where everything sort of comes together. But you're right. I mean, I think back to, because I was involved in Applied Biosystems when we did the genome, and wow, that was such a big deal. And then every once in a while, I still see an article saying, yeah, the genome hasn't really done. And I'm like, are these people how do they write these things with any straight face? And it gets published in a reasonable journal. And I'm like, these people are out of their minds, considering everything in biotech, everything in, functional genomics, all this stuff is grounded in that information.
Right. Oh, I see the same stuff and I think, what do you mean nothing has come? What are you talking about? Everything has come from that. And then, when I point to success stories with individual patients, which are growing and growing, they're like, yeah, but that's the exception. Well, it's the exception that's turning into the rule.
Yes.
It's more and more. And I think what you're seeing now is that, with any new technology, the barrier to entry starts very high. But that barrier has been falling fast to the point where, people who start off on, the parent side, like me, are increasingly finding that they can get into the game and that they can do something. And I think it's at a level now where almost any, patient or parent that has a technical background can jump over and do something. But even patients without that background are making the jump now too. So the barrier to entry is falling so fast that it really has changed everything when it comes to, patients moving the needle for themselves using the fruits of the genomic era.
Yeah, and I think computational, power and cost and ease of use are starting to come down dramatically, which then, brings the two together, which is, of course, the like sort of the idea of behind the whole show and everybody that I talk to. And I see some of the companies I talk to, they're like, yep, we sort of eliminated 3 years of work. We could get it done in, a week to 2 weeks because of what we're looking at, how we've applied our computer science, and how many new pathways we can sort of identify, of course,
Right.
for new drugs.
Yeah. I can give you examples of where the barriers fell overnight as I needed them to, just by luck. For when it came to creating model organisms, right before CRISPR, gosh, that was an expensive, daunting process and took a lot of time. And then CRISPR shows up and they're like, oh yeah, no, it'll be a few months and $10,000. And it was, just like that, it happened. And there's equivalent revolutions happening on the computational side too. If you look at, protein folding technology, it was a joke, that like, yeah, it'll never happen in silico. And then all of a sudden, like now maybe the only way we'll ever get some structures is in silico.
Right.
And then that was
Right.
kind of an overnight thing too. Obviously it wasn't overnight for the engineers on the project at Google. But once it appeared, it's like, oh my gosh, what a game changer.
Well, and then as soon as somebody does it, it motivates more people to sort of and it sort of moves the space forward that much faster. That's the part I find interesting, is most people have trouble understanding the speed of change. And it's moving faster now than, and I'm used to, trying to keep track of how fast everything's going, and I'm finding myself having trouble keep up with how quickly things are shifting.
It really is changing faster than I think any one person can predict. And the disruptions are coming, almost out of nowhere. Like no one saw CRISPR coming. You might reasonably foresee that at some point, some efficient gene editing technology would have emerged. But I think it emerged much faster than was expected. I remember when I would work with patients, 5 or 6 years ago, I'd say, yeah, there's this thing, these antisense oligonucleotides, and maybe someday, but we're probably, and I would say at the time, like maybe 20 years away. You see oligonucleotide therapies really take off. And, then I think it was 2 years later, there's an FDA approval. And then a couple of years after that, there's the first big N of 1 introduction. And then like a year and a half later, we're all injecting mRNA into ourselves. Like, well, that happened pretty fast. It wasn't 20 years.
Yeah, and for people like you and me that are in this, like, my mind is like, right? I'm like, wow, this is awesome. And then I try to explain to someone and they're like, they don't understand like the impact that some of this is happening and the implications of what we're talking about.
Yeah, and I think that going forward, it's going to be a much steeper acceleration than anybody can really predict, because we've suddenly just burst into the era of programmable therapeutics. And I mean, COVID really, it suddenly just threw it on the table. There it is.
Yep.
And say, and an example as well, people say, okay, well, if you can just give mRNA directly, and well, instead of trying to deliver these complicated proteins to do the gene editing, why don't you deliver the mRNA for the CRISPR protein, or for Cas9 and in its derivatives, along with the guide RNAs? Well, that's much easier, and my gosh, it looks like it might actually work. So these things, they couple in unexpected ways, and very quickly too. And so I'm excited because I have no way to know what's coming now.
I've always felt I don't know what's coming. That's why I try to read such a broad array of sources, everything that's going on in IT and chip development to what's going on in our world. But I think the next big wave of shifts is going to be how a lot of this gets implemented, the business models behind it, and that's the next big shift, because you don't have to do it exactly the same way you had been doing it up till now.
Oh, I agree. And oddly enough, I spend a fair amount of time thinking about, stuff as mundane as how do we get payers to actually pay for some of these things? How do we show them that there's value to be captured already? And because there is, I think we're not far away from a future where payers realize it's going to be cheaper to take this very expensive patient with a complex disease and look for sort of a root cause treatment than to continue paying for symptomatic treatment. I think we're at the threshold of that era.
Well, I think, the CEO of Illumina said we want to get whole genome down to $60, right? I mean, at some point you're like, okay, when are you gonna stop being worried about the cost of this? Because it's so, I mean,
Right.
it's going to be a rounding error at some point.
Yeah. And if you
That
think about it over the course of someone's life, it's already a rounding error.
yes,
It's already there. It's already
but
below $60 a year.
$60. Yeah, I mean, I was talking to a company where they could do, if you could do the initial analytics for 60 and then do the computational on top of it for another 60. At some point you're like, look, we should just be doing this for everybody. The problem is the implementation, and can physicians keep up with what
Right.
does it all mean and what am I supposed to do?
Yeah, and that's why I think, when I talk about precision medicine and data as a drug, I always have to highlight the importance of computational aid for the physician. Because if you were to give a physician a raw genome, they would go, "What? It's a paperweight, it's a hard drive. I don't know what to do
Right.
with that." Even if you distill it down to the individual mutations, the average physician goes, "I still don't know what to do with that." It's got to be broken down into something far more actionable for them. And I think we're going to look back now as sort of like the dark ages of IT and medicine, because we're in a situation where I don't know any physician that loves the EHR they use. They in fact, they all hate it. It is a disastrous user experience across the board. Yeah, and this is, it's like a classic problem in software where the people who pay for the things are not the people who use the things.
Right.
So what are EHRs optimized for? Billing.
Yes.
There's only one EHR, as far as I can tell, that's optimized for patient care, and that's at the VA, where they're not really concerned about billing. And so people like that one, which is, not a big surprise.
Well, and they were talking about, they wanted to put in Epic over what got built, and I was like, who got paid like behind some closed door to make that decision? Because I'm like, that was the dumbest decision I've ever heard anybody make.
I thought the same thing, having worked on the Million Veterans Program. I'm like, no, that's the crown jewel. That thing actually works, and it works well. And it gets great data. Do not replace that. Keep it as is.
Yeah, well, you need to have, unfortunately, whoever's making that decision has no, skin in that game, as far as I can tell. But I agree with you. I mean, it was, I've said over and over, if anything's going to break medicine, it's going to be the existing EMR systems, because you can't innovate if you can't get the data out.
Right.
And Google and Microsoft and Apple and everybody's innovating because they get to change their system at will, right? Everybody gets to jump on AWS and innovate. The system is sort of stuck in stasis and can't move out of it, which is what I find worrisome. But
I agree. You've either got to get the data out or the computation in, or preferably both. I've dealt with physicians where I can say, hey, we could give you this really cool genomic test for your patients, and then if they try to take a drug, you'll know if it's not going to work for them. And they go, well, will there be automated decision support in the EHR to tell me if that happens? Or do I have to sort of look at the note and see that they have this variant? I go, well, you're going to have to look at the note. I said, no, I don't want that. I do not want that, because if that note is in there and I don't figure that out and I prescribe a drug that causes an adverse event, I'll get sued. But if the information's not there at all, I can't be sued. I mean, that's the world we live in.
Wonderful. Well, listen, it was great to speak to you. Some of the stuff you're doing is awesome. I wish more people knew about it. I wish more students were involved in it so they could get firsthand experience, because like you said, I think that's when we can start to teach people the crossover between, medicine and computational work in general. Because I'm always trying to find people that know both, and there's not a lot of fruit on that tree at the moment. More is growing, but not as much as you'd like.
I agree. We need to get people going more often in both directions. And that's, that's one of my missions at the Institute as well, is to cross train folks into both sides, biology and computer science.
Excellent. Well, it was great to talk to you. I appreciate the time.
Likewise, it has been a pleasure.